Hello, my name is Justin Duffy, and this summer I worked in Dr. Dwayne Townsend's lab. My project investigated the effect of mast cells on the physiological progression of Duchenne muscular dystrophy. Duchenne is the most common and severe type of muscular dystrophy, but the exact mechanism of muscle degeneration during the disease is not currently known. One hypothesis is that mast cells regulate the development of Duchenne. Mast cells are primarily an immune cell, but they've also been implicated in the development of fibrosis and muscle degeneration in other disorders. My study investigated the quantity and localization of mast cells in the hearts, quadriceps, and diaphragms of MDX mice, which are an animal model of Duchenne. I observed a significant increase in the quantity of mast cells in the MDX hearts and quadriceps, which supports the hypothesis that mast cells regulate the progression of Duchenne. However, the lack of a mast cell increase in the MDX diaphragms, in addition to differences in the localization of mast cells in all three tissues, suggests that mast cells exhibit a tissue-specific response in Duchenne muscular dystrophy. My study also observed an increased concentration of mast cells in the serous membrane surrounding the hearts and diaphragms, suggesting that mast cells may use this membrane as a means of locomotion through the body.
Hello, my name is Gabrielle Evans, and I was in the Thomas lab this summer. I focused on the Ryanodine receptor, which regulates calcium release. This calcium release is essential for muscle contraction. In fact, dysregulation of the Ryanodine receptor involving a leak state is associated with several diseases such as heart failure and muscular dystrophy. This is why it's an important target for finding inhibitors of that leak state, which is what my work was. I used fluorescence spectroscopy to measure the effect of various compounds on the structural conformation of the receptor, and I found several compounds whose readouts indicated a closed state of the receptor, and my work is one step in a longer process of identifying compounds for the treatment of RYR-related diseases such as heart failure and muscular dystrophy.
Hi, I'm Shannon Flannery, and this summer I had the pleasure of working with the Kawakami lab to investigate the role of the C/Cl12 chemotactic gradient in zebrafish hearts. Zebrafish are a small fish native to India that possess the unique ability to regenerate their organs, most interestingly, their heart. The C/Cl12-CXCR4 chemokine gradient regulates how cardiomyocytes migrate to the wound area and proliferate. I looked at how negating the C/Cl12 gradient would affect cardiomyocyte proliferation. I negated the gradient by overexpressing the homing ligand and a decoy receptor. We found that when the gradient was negated, proliferation increased. This suggests that C/Cl12 plays a role in promoting cardiomyocyte proliferation as well as the creation of a gradient. We hope that one day this research can be applied in human models to prevent the formation of scars after heart attacks.
My name is Jeremy Heinley, and I worked in the Kaiba lab this summer. My project dealt with attempting to find new markers that can identify myogenic progenitors from the cardiopharyngeal mesoderm, or CPM. The CPM is important in development because it is the only known tissue that can develop into either cardiac or skeletal muscle. Its development is still poorly understood, and in order to identify this tissue from the surrounding tissue, surface proteins are used. Only two are known that can separate this tissue, and they are PDGF receptor alpha and VCAM. We used the technique fluorescent activated cell sorting to potentially identify new markers that can identify the CPM. We tested 12, and we found that 3 alpha-7 integrin, Notch1, and PDGF receptor beta can potentially identify the skeletal muscle lineage, and CD44 can potentially identify the cardiac lineage. Future research will have to be done to see if they can actually identify the CPM lineage, and hopefully they can be used to further purify and identify this population.
Hi, my name is John Holm, and I worked in the lab this summer studying Duchenne muscular dystrophy. Duchenne muscular dystrophy is best known as a progressive muscle wasting disease, but it is also characterized by an increased prevalence of anxiety disorders. Similarly, the mouse model for this disease, the MDX mouse, suffers from an exaggerated stress response that is shown by increased glucocorticoid levels and a physical inactivity response in response to a mild stress. To investigate the role of the HPA axis in the generation of this response, we inhibited it at several points with several drugs to see if we could stop the inactivity response. We found that this did not stop the inactivity response, and based on this, we've concluded the HPA axis is not involved in the generation of anxiety in Duchenne muscular dystrophy. This is important because it opens the doors for other potential avenues of research, such as looking into the sympathetic nervous system or potential inflammation-driven anxiety generation in Duchenne muscular dystrophy patients.
Hello, my name is Kevin Janisek, and I worked in Dr. David Thomas's lab this summer. My project was about functionally validating drug screening methods that targeted both RYR and SERCA. So RYR and SERCA are important in the calcium cycling that's needed for muscle contraction. When this calcium cycling becomes dysregulated, then pathologies such as heart failure and muscular dystrophy can occur. So our overall goal was to find therapies that would target RYR and SERCA and hopefully fix this dysregulation. So specifically for my project, I looked at the functional effects of various drugs individually on RYR and SERCA. So overall, we found 3 drugs that impacted RYR and 4 drugs that impacted SERCA in a way that would fix this dysregulation.
Hello, I'm Anders. I work this summer in the Tranquilla lab. A little bit of background about my research project. Chronic venous insufficiency, CVI, is a commonly occurring condition in which the venous valve fails and begins to leak blood. As this leaked blood pools in the victim's legs, it can cause strokes, ulcers, and even require amputation. My research was focused on evaluating a tissue-engineered venous valve, the TEV, grown in lab, versus native jugular venous valves from a sheep and a pig. I designed a mounting system to secure the valves for testing, while I used a video camera to record video and a LabVIEW program to record data. The leaflet motion and pressure drops of the TEV compared favorably to the native valves, thus suggesting the TEV has high potential as a venous valve replacement, allowing us to fulfill a critically unmet clinical need for the 7 million plus CVI patients in America. Ultimately, my research provides an effective method of testing TEV prototypes before implantation, not only allowing us to limit the numbers of animal sacrifice, but also allowing us to streamline the TEV development cycle.
Hi, my name is Tom Kane, and I worked in the lab of Dr. Michael Kaiba this summer, investigating the process of skeletal muscle regeneration. Skeletal muscle has a very robust ability to regenerate itself in response to things such as injury or similar types of trauma. And the physiological basis of this is a population of stem cells called satellite cells. These cells exist in muscle tissue and in response to trauma can proliferate and eventually differentiate into new myofibers. My project specifically investigated a potential modulator of this process, the cytoplasmic receptor aryl hydrocarbon receptor, also known as AHR. To investigate whether or not AHR impacts the process of muscle regeneration, we generated an inducible knockout mouse model, which upon the administration of the drug tamoxifen could no longer express the AHR protein. Then injured a specific muscle group of that mouse with the compound cardiotoxin in order to determine whether or not the process of regeneration was impacted by AHR's expression. In terms of results, we found that AHR is most likely necessary for normal muscle regeneration, as the injured muscle group of mice lacking AHR expression weighed significantly less following regeneration. More specifically, we found that the cross-sectional area of individual myofibers of these injured muscles was significantly less. In total, the results from my research indicate that AHR is necessary for normal muscle regeneration, and therefore opens the avenue for further lines of research investigating the regulators of the process of skeletal muscle regeneration as they relate to AHR. In conclusion, the results of my research indicate that AHR is necessary for normal muscle regeneration, and therefore it lays the foundation for further research to characterize the process of skeletal muscle regeneration as it relates to AHR.
Okay, so I'm Karina, and I worked in the Perlingero lab this summer. So previously, the Perlingero lab developed two lines of what we call inducible myogenic progenitors, which basically are developed from stem cells, and they will become muscle precursors when cultured with doxycycline. And these progenitors have a very interesting or special property that when they are injected into the bloodstream, they will engraft in injured skeletal muscle, which is different from satellite cells, which are the adult stem cell population that would need to be directly injected into the injured muscle. So for my project, I looked into the genetic mechanisms of this property. And so I used basically a lot of bioinformatics analysis to identify genes that were highly upregulated in the myogenic progenitors that we developed in comparison to the satellite cells, which don't have that property. And then I was able to verify the expression of some of these genes using qPCR and immunofluorescence staining.
Hi, my name is Megan Noworiak, and this summer I was in the Visible Heart Lab led by Dr. Paul Izio. My project focused on creating a 3D printed reusable heart model that can be used by surgeons to simulate a procedure called ECMO initiation. ECMO is a type of bypass life support that can be used in infants born with heart and lung problems. The basic ECMO circuit involves removing venous blood from the patient, reoxygenating it, and then returning it to the arterial system of the patient. To connect an infant up to this ECMO circuit, we typically utilize vessels in the neck, or a surgeon can go and surgically open the chest and cut into the heart. To help surgeons practice this difficult procedure, I created a 3D printed reusable heart model that can be used to practice this procedure on. My final model was watertight and flexible, so artificial blood could be run through the model during simulation, as well as CPR and cardiac massage. Overall, hopefully this model will be used for ECMO initiation scenarios for hospital staff to practice and hopefully be better prepared for real ECMO situations when they do occur, and reduce the occurrence of cannulation complications when it comes to neonatal ECMO.
My name is Sarah Romans, and I worked in Dr. Brenda Ogle's lab this summer. Previous research has shown that some cardioactive drugs can affect epileptic activity in addition to cardiac activity, so we wanted to investigate the intersection between cardiac dysfunction and epilepsy. Cardiomyocytes derived from stem cells can be a good model for how the human heart responds to different drugs. So we decided to assess cardiomyocyte response to 5 different drugs that have previously been studied in order to verify that our methods produce the expected results for these drugs. We did this by tagging stem cell-derived cardiomyocytes with a fluorescent calcium marker and imaging their beating under a microscope. And in doing so, we were able to show that our methods produce the expected results for these 5 drugs. Our next step is to apply these methods to stem cell-derived cardiomyocytes with a mutation in the PCDH19 gene, which is commonly associated with epilepsy, to see if these cardiomyocytes respond any differently to cardioactive drugs than wild-type stem cell-derived cardiomyocytes.
Hi, my name is Mega, and this summer I had the opportunity of working in Dr. Joe Metzger's lab. Stem cell biology is a very promising area of research, and stem cell-derived cardiomyocytes are supposedly accurate models of adult human cardiomyocytes. However, the lack of CTNI, or cardiac troponin I protein, makes these cells not very accurate models. The goal of my project was to increase CTNI levels in these stem cell-derived cardiomyocytes by inhibiting a molecule called antisense CTNI, RNA. In doing so, we were able to increase CTNI levels by a very minuscule amount, but with further trials, we will be able to increase CTNI levels through more targeted methods. Applications of maturing cardiomyocytes include disease modeling, drug testing, and regenerative therapy.
Hello, my name is Marta Williams, and I will be a junior at Luther College. This summer, I worked in the Visible Heart Lab under Dr. Paul Izio, studying the risk of tricuspid valve damage during the process of micro-leadless pacemaker extraction. Currently, microdevice extraction involves pulling the exposed microfixation tines through the tricuspid valve before sheathing them into a cup introducer. In my project, I engaged single chordae of swine heart tricuspid valves with 1 and then 2 microdevice fixation tines and retracted them using a force machine to measure the force produced by microdevice extraction. I also engaged single chordae with a metal bar to determine the force required for chordae rupture. My results indicated a statistically significant difference between the force required for tricuspid valve chordae tendinate rupture and the force produced by microdevice extraction. I also found significant anatomical differences in both the magnitude of forces required for rupture and the location of rupture for primary and secondary chordae, respectively. These results demonstrated that there was a relatively low risk of damage to the tricuspid valve due to micro-leadless pacemaker extraction in healthy swine hearts, suggesting that these procedures are likely to be safe in regards to maintaining tricuspid valve integrity. However, further experiments should be done using human specimens to verify these results.